Hello all, Konnichiwa. I welcome you all today for today's webinar, Unlocking New Therapeutic Uses, the Role of Drug Repurposing in Modern Drug Development. Today we are going to talk about repurposing and its uses, how it came into being, the methodologies we use at Excelra for the repurposing, and advantages for engaging with Excelra for this solution. So we all know that drug discovery and development has been a very cost-intensive and time-intensive process. For a drug to come in the market, it takes nearly about $2 billion and 10 to 17 years. However, after so much of investment as well, the success rate of any drug to be launched into the market is fairly less, about less than 12%. If you look at the table on the left, it showcases the different drugs introduced by companies over a period of years and the associated cost with each of those drugs. If you look at the graph on the right bottom, it shows the success rate of each of these drugs moving to the next phase in the clinical trial. For example, from phase 1 to phase 2, only 64% of drugs are successfully moving ahead in the clinical trial. And as we go ahead, the success rate goes down to 32%. So overall, what are the reasons why these drugs fail during these transitions to the next phase of the clinical trial? The answer is in the graph on the top right-hand corner. The main reason is the efficacy. Around 54% of drugs fail in the efficacy and hence fail to move to advance to the other levels of clinical trials. So what is the solution on this grave problem where you invest so much of money, so much of time, but you cannot expect good returns? So repurposing is a good way of rescuing your assets, your investments, and your time. Repurposing gives a new chance to the drugs to come into the market for another indication or another drug for a particular indication. And this new lease in life can help them to rescue the millions of dollars invested in research. If we look at traditional drug development, which is in the top half of the slide, you can see that the cost of investment of any drug development is $2.7 billion, and it takes around 10 to 17 years for it to come to the market with a less than 10% probability of success. Each of the different stages of drug development, from target discovery all the way to development and registration, requires intensive amounts of investment in terms of time and money. If you compare this with drug repositioning, there is substantial saving in costs in terms of the money as well as the time. Also, these drugs have proven safety and pharmacokinetic properties, which makes it an attractive option. It usually begins with identification of the compounds, its various uses, acquiring those compounds from relevant companies, development and registration. So it is a viable and a very good option in terms of rescuing your assets and the investments in research. Now let me talk a little bit about the methodologies we adopt here at Excelra for repositioning. I invite my colleague Akshay to talk more about these methodologies.
So thank you very much, Tulika. If you look at the repurposing methodologies that are out there, so they can be broadly classified into two different varieties. The first one would be a literature-based methodology, and the other would be an algorithm-based methodology. As you can see in this bar chart, in the last decade or so, there has been a massive upsurge in the number of publications that are being published in the field of drug repurposing. Now, a scientist can leverage the data that is being published here to understand the various facets and can use this for the drug repurposing exercise. So this is the literature-based drug repurposing methodology. The other methodology that we can do is based on algorithms. And here in the next couple of slides, we are providing a couple of examples of algorithm usage in drug repurposing, which also involves a lot of artificial intelligence and machine learning models to sort of score and to rank the indications. So the first algorithm that we are going to talk about is structural similarity. So this algorithm is used to identify similar compounds as like the input drug, so that they can have a similar mechanism of action or they can have similar activities. Right. So in this case, we employ two different processes: the 2D similarity and 3D similarity. So when we do this, we run our compounds against various databases which have a library of compounds available to them and are used to create the fingerprints and score. So what we do is we take a particular compound and then we do a 2D similarity using different compound fingerprints, and then we have a list of an output compounds which 2D similarity score. And then we then select the list of from the list we select the compounds with greater than 0.8 Tanimoto score, and then we use that again to shortlist the compounds which are then again used for 3D similarity. And similarly, compounds with greater than 0.8 score are then shortlisted and the indications are associated with the output drugs. Here, to calculate this the Tanimoto coefficient, which is a very useful and important index, we use various AI and ML models. The next approach that we use in the algorithm and is quite regularly employed is the drug-drug gene signature methodology. Here we take the gene expression, the differentially expressed genes from both the case and control, and then differentiate them into up and down regulated genes based on their expression patterns, fold change values. These are then fed into an algorithm, which then classifies the gene signature based on a similar gene signature or an inverse gene signature. Again, these gene signature patterns can also be employed to identify alternate drugs or alternate indications. Again, several AI models such as statistical analysis, etc., have been used quite regularly to shortlist these drugs. So the basic principle is that the drugs with similar gene signature might act in a similar fashion as the approved or clinical trial drugs for a particular disease. So at Excelra, what we have done is we have brought together the best of both these methodologies and have created a process which is quite useful to all our partners. So it has three different aspects that are mentioned here. The first is that we have a custom database which is called the Global Repurposing Integrated Database. This database contains a few proprietary databases that are part of the Excelra, as well as several 40 plus public databases. The data is all captured and fed into this particular database. Apart from this, we also have a set of repurposing algorithms. Some of the algorithms that I had mentioned in the previous slides. So we use this information. And the third is the analytics engine, where actually the subject matter experts use this information and help actually draw those particular connections and come to final conclusions. These are then presented to clients in forms of various visualization or as part of reports that we will be talking about in the upcoming slides. So basically, the four pillars of this are that we talk about the drug disease target associations, we provide biological insights, we provide also inputs on testable hypothesis, as well as we provide details to clients if there are any animal models existing for them to conduct their proof of concept studies. So broadly speaking, the repurposing methodologies can be classified into three types. The first type is type of uh, input. The second is type of approach. And the third one is type of analysis. And in the upcoming slides, we will be discussing one by one these different methodologies and how we have successfully delivered various projects to clients employing these approaches. So we will begin with the different types of approaches, inputs, sorry. So when we start with input-based repurposing, the input can either be a drug, it can be a target, or it can be a disease. When we use drug as an input, the focus is to identify alternate indications. When we take target as an input, we can identify either alternative drugs or diseases as the outcome. And when we take disease as an input, we can identify again either alternate drugs or targets as an input, right? When we use these different repurposing approaches, we can also, when we use drug-centric repurposing, we can look at either generic drugs or innovative drugs or marketed drugs or failed shelved compounds. There is also a possibility to look at combination partners. When we have a disease-centric repurposing exercise, we can have a normal disease-centric repurposing. We can also have a focus on rare and neglected disease. And then we have, when we use a target-centric approach, we can use any single target as a customized target as an input and use it for downstream analysis. So I'll give you an example of each of these different approaches in the next few slides. So we start with the drug-centric repurposing. So typically what we receive from the client is a single drug. And then we also receive the target of the drug. And the idea is that we push this drug through this multiple algorithms that we are employing within the within Excelra. So this then provides you with a list of indications. This is stage one. Then once you have the list of indications, you then arrive at a final list of indications, and then based on certain criteria such as the client's interest as well as the high level of scientific rationale, we then go to a detailed analysis in stage two, where we prioritize the diseases and then provide our analysis based on the strength of evidence, the mechanism of action of the drug compound versus the disease pathophysiology, and also how easily we can design the proof of concept studies. So in the next few slides, I will talk in detail about stage one and stage two. So when it comes to stage one, you input the compound target pair into the algorithms, and then this is then uh, uh, we receive a list of filtered drug disease associations. Then these drug disease associations are standardized by using certain ontologies, as well as by removing general indications, as well as unifying certain anonyms and aliases. So finally we get a frequency table, a snapshot of which is pasted here. And this frequency table then lists the different approaches from which the particular indication has been obtained, and there is a ranked order of these indications. And using then different methodologies, we then shortlist the indications. Then after discussions with the client and different, looking at the available evidence, we move to stage two, where we perform customized deliverables for these indications, which includes making detailed analysis of the mechanism of action, the target role in pathophysiology, the strength of evidence, disease model recommendation, the identification of biomarkers if necessary, patient stratification strategies, and we also provide custom pathways which show how a particular drug might act on the disease pathophysiology. This is the second one where we have used a disease-centric repurposing approach. So here the objective is to identify drugs for a particular disease of the client's interest, right? So again, we looked at this in this particular project. We shortlisted 5 drugs, and then we also provided the experimental design as well as proof of concept studies, information to the client. So again, we use the disease of interest as the input, and then we go through the multitude of approaches. And then in the end, we come to the list of drugs, which are again prioritized. The diseases are prioritized based on various methodologies, various criteria, which are listed by the client, as well as discussions between us and the client, and the high level scientific rationale. So this particular slide gives you a snapshot of the custom pathways that we are providing. So this particular indication was a dermatological indication, and the 5 diseases that we have proposed here, you can see the MOAs for this, and we have looked at the different pathophysiological processes that are sort of used by these different drugs to are blocked by these different drugs. So this provides a very good scientific rationale also for using these different drugs. Next, we come to the target-centric mechanism. So again, here target-centric mechanism was that the input was stress-responsive transcription factors, and the client was particularly interested in identifying kidney disease, kidney diseases. So, and then we looked at various indications, and in the end, the client chose 2 indications, which were for preclinical testing. So like we said, again, we use these different algorithms for our hypothesis generation. And then when we got a list of diseases, these were then, again, after discussions with clients, we ruled out cancer because it was not of their strategic interest. We also ruled out neurological indications because the compound had low brain barrier penetration. So when we looked at, certain inclusion criteria. So after these various criteria, again, a list of diseases were drawn, 6 diseases, and then a further deep dive was performed, as mentioned in the earlier slides, to generate testable hypotheses. So these are the few examples of input-based repurposing. To explain the other slides, I will again request my colleague Tulika to please explain the next few slides.
Sure. Thanks a lot, Akshay. I will now be talking about approach-based repurposing. So essentially, this methodology of repurposing combines the in-house data which is generated by the clients with our in silico approaches which Excelra adopts for repurposing programs. This makes the repurposing programs more robust and our recommendations more tangible for our customers. If you look at this figure on the left-hand side, you can see the in vitro methods with which data is generated by customers. They could be high throughput screening studies in the type of phenotypic assays, or there could be omics data which is being generated in the form of genomics data sets, transcriptomics data sets, proteomics, metabolomics, or in certain cases, we have also received lipidomics data sets. What we do with these data sets and these phenotypic screening assays is we take the data and we analyze it. We analyze the data to identify differentially expressed genes, proteins, or metabolites. And we utilize the phenotypic screening data to understand the phenotypic impact of the drug or the compound in question. We then use our in silico approaches in the knowledge-based and computational methods. In the knowledge-based methods, we use different methodologies to identify alternative indications or alternative drugs. We utilize literature mining by scanning the different scientific articles, patents, etc. We utilize clinical trials to identify the clinical landscape of the various compounds or the diseases. Biomarkers are identified. Various approved drugs or current existing standard therapy is noted. As Akshay mentioned earlier, we also utilize a lot of gene signature data to identify diseases with similar gene signature or drugs with inverse gene signatures. Now, this data can also come from the omics studies, that is the in-house data which is generated, which I talked about earlier. We also do a lot of structure similarity, adverse events, and many other approaches as have been enlisted over here. We then utilize the data, which is basically the drug target and disease relationships identified to build network models. These models are basically connections between the different targets or genes overlaid by pathways and the outcomes. Most of these connections that are identified by the knowledge-based approaches are then validated by public data, and then we utilize machine learning models or predictive models to do predictions for the different diseases that could be potential repurposing options. So this is what we essentially do in approach-based repurposing. We combine the wet lab data from the in vitro studies, which is mostly provided by our customers, with our in silico approaches in order to make our recommendations more viable. So this is an example of a similar approach where we have used integrated drug repurposing. This was done for 6 compounds which were at various stages of their drug development journey. Some of them had failed in the clinic, some of them were in the phase 1 terminated asset, some of them were discontinued because of safety or toxicity reasons or business reasons. So these drugs were then, the repurposing program was utilized for identifying alternate indications for these drugs. We were provided certain omics data by our customers. This omics data was basically from the in vivo and in vitro experiments. And this data was then analyzed by our subject matter experts to identify differentially expressed genes and differentially expressed metabolites. We also used various in silico methodologies, as I had discussed in the earlier slide, the knowledge-based methods to identify the different alternate indications for these 6 drugs. This was how the results looked. We identified certain indications based on our in silico analysis, and we also saw whether the signatures identified by the analysis of the genomics and metabolomics data were identifying similar diseases. And this is how the diseases were prioritized. We found several cancer types, neurological disorders as well in this particular analysis. And in this way, we prioritized 4 diseases which were showing coherence between the in silico analysis and omics analysis. So in this fashion, in integrated drug repurposing, we basically combine wet lab data with the robust scientific approaches for extracting data from the public domain, as well as analyzed data from the different data sets in the public domain. This makes the repurposing exercise more reliable. Now, let me talk a little bit about the analysis-based approaches. Analysis-based approaches are basically high-throughput screening exercises where we begin with a list of drugs or a list of diseases. Till this time point, both me and Akshay have discussed the different approaches where we can begin with a single disease in mind to identify alternate drugs, or a single target and a single drug in mind to identify different diseases. But what if we have a list of different compounds or list of different drugs or diseases in mind? These are some of the examples where we can begin with more than 100 drugs and identify alternate indications for those. In this particular exercise, we had a list of 123 drugs and their associated targets. As Akshay mentioned, we have a proprietary global repurposing integrated database which combines data from 40 plus public databases. And we utilize these different algorithms showcased here to identify drug target and disease relationships from the database. Ultimately, we end up with a table as has been shown here, and this table showcases the drug and disease relationships and the associated clinical phase. As you can imagine that for 123 drugs and the identified indications which were associated with them, the data was very large. And hence, to understand the data, to navigate through it, We built a visualization tool which can be an easy tool to navigate these relationships and understand the associations as well. With the help of these visualization tools, our customers could identify certain drug disease pairs of interest to them and which was strategically relevant for their organization. And then we built a testable hypothesis, which is like a business report, which can help you pitch the drug disease pair identified to the management. This is how the visualization tool looked like. It had a disease page, which was like a heat map. Each of the square represented a particular disease, and the color represented the number of drugs that were identified for the disease. If you clicked on the particular disease, you would be diverted to the drug page, which is at the center, which has the different drugs associated with that particular disease. You could also click on one of the individual drugs and then identify the different diseases the drug was associated with. So it was an easy tool to navigate and understand the different associations between drug, disease, and target. So this was an example where we began with a list of drugs and we went and identified a large number of diseases associated with them. The second example is when we begin with a list of diseases and then identify a list of drugs which would be relevant for them. This particular exercise was done for the CNS indications, which is a difficult area to work with. We began with a list of FDA approved CNS drugs and clinical trial compounds which were associated with the CNS indications. A matrix was created of drugs versus diseases, and then we prioritized these drug and disease pairs based on certain criteria. We looked at diseases which had a large number of papers published on them, that is, which were more than 10 in number, which were prioritized at low priority. We also looked at indications where there were less than 10 publications, which was categorized as medium priority. And then finally, we identified indications where there was no association associated with these indications. No FDA approved drugs, no clinical trial compounds, and no papers published. So these were of highest priority to the customer. They wanted to basically identify indications and disease pairs where there was no association between the drugs and the diseases. In the second milestone, we then looked at each of these drug and indication pairs which were identified via various methodologies as has been put together in the box in between. And finally, we shortlisted drugs and disease pairs which were most relevant for the partner. This was how we did the filtration exercise or prioritization of the drugs which were identified. We began with 243 drugs in total, and then we shortlisted 165 high priority drugs, that is, drugs which did not have any published association between the two. Then we shortlisted 87 drugs based upon the strategic interests of the customer and the different methodologies which had been enlisted in milestone 2. On deeper analysis, we could look at the target association of these particular drugs with the alternate indication. In such a way, we could identify 53 CNS drugs corresponding to the 103 drug disease pairs. This is how the recommendations look. If you look at the diagram on the right, you can see the different CNS indications covered within this exercise and also the number of associated drugs for each of the indications that was identified. So this is another example of how you can begin with a large list of either compounds or diseases and identify alternate indications for them. This is an extremely high throughput methodology and saves a lot of time and covers an entire therapeutic area for recommendations. There are several validation and success stories for our methodologies in the public domain. We have done several different projects with various Japanese customers as well. And as showcased by me and Akshay in the previous slides, we cover different aspects of repurposing which are most suited for our partners. This could be various input-based approaches where we begin with either the drug, target, or the disease. It could be high throughput exercises where we begin with a large list of either the drug or the disease, or it could also be combining the in-house data, which is the wet lab data, and the in silico methodologies within Excelra. And the testament to our approaches is with all of these validation stories, which are in the public domain for you to have a look at. We are also not very fixated on the different indications or therapeutic areas we work with. We can work with several different therapeutic areas like oncology, inflammation, neuroscience, cardiovascular disease, immunology, rare and orphan diseases as well. Most people are quite wary of rare and orphan diseases, but we've also worked in areas where there are less number of publications available, and we can get the best out of that data for recommendations. Our 100 plus talent pool of different individuals having advanced degrees in chemistry, biotechnology, drug discovery and development, and pharmacology help us address all the needs of these different kinds of projects. We have successfully delivered more than 100 plus projects, and we have 10 plus years in experience in repurposing. This is testament to the fact that we have given actionable insights to more than 16 plus top pharma companies across the globe. So this is how we navigate through various repurposing programs at Excelra, and we provide actionable and relevant insights to our partners. So I thank you all for the time today. Arigato gozaimasu. And we would love to hear from you, feedback, as well as if any questions you have. Thank you.
Thank you.